Very good. So the chapter 10, it's about the replication. Okay, so we will discuss the replication. So the replication of DNA. Basically, replication that will occur, DNA replication will occur right before the cell division, because you need to replicate the genomic material before they perform the cell division. And for the genetic information, right, the flow of the genetic information, let's say this is the parent cell, and then inside the cell, this DNA can perform the transcription, translation, that's gene expression, okay, that depend, and so cell can metabolize and grow. That's a gene expression, which we will discuss in chapter 11, 12. And also the genetic material can also perform the recombination, all right, recombination with another cell. So basically for the bacteria cell, the recombination happen during the transformation, transduction, and conjugation. And also the DNA can perform the replication. So that would be during the cell division, right? Before the cell division, they have to replicate the genetic material, and therefore they can equally distribute to the next generation. So that's the DNA replication. And the DNA replication, so basically in the early days, there are several different hypotheses. So they have this conservative hypothesis. So conservative hypothesis mean after the replication, then the original DNA still remain a double helix with the original all the parental DNA, and the newly synthesized DNA, they will form a double helix by themselves. That's the conservative replication. And then semi-conservative replication, that means after replication, each double helix will be a hybrid of the newly synthesized DNA strand and also the parental strand. And dispersive just randomly mixed together with the new and newly synthesized DNA fragment and also the parental fragment. So dispersive. So in order to identify which the mechanism is the correct DNA replication mechanism, then there's a experiment to approve the semi-conservative replication. So basically, in the experiment, is they use the isotope label the nitrogen, right? And then to be the only nitrogen source to feed the bacteria, okay, to feed the bacteria, and then I think it's here, right? So you have the bacteria cell, all right, and then you grow in this isotope label nitrogen 15. This isotope label the nitrogen source, so that this nitrogen, remember the DNA structure. We have the nitrogen in space, sugar and phosphate. So this isotope, the heavy isotope, heavy nitrogen, then will be incorporated into the DNA. All right, and then after they make sure that all the DNA carry the heavy nitrogen, then they switch the bacteria cell into a media that contain the regular nitrogen 14. So after one generation, then they collect the DNA, they isolate the DNA, then they try to see the composition of the genetic material, okay, and then so they collect the after one generation, they switch to the regular nitrogen source media, then they purify DNA and then apply the centrifugation, all right, and then the first generation, of course, the you should see the all the DNA are the heavier nitrogen, and nitrogen 15, okay? And then the second generation, so if it's the conservative, all right? So you have N15, N15, and then after one generation, all right? So you should have the 15, 15 and 14, 14. Or you can have a semi-conservative. That'll be the mixture of one band of the 14, 15 mixture, all right? Or for the dispersive, can also be the 14, 15 mixture. Only this conservative will show you two different bands, but for semi-conservative and dispersive will show only one band because it will be the M14, 15 hybrid, okay? So, and then they perform these experiments. So and then they see the first generation, all right? After the first generation, they see they can only see one band, all right? And so that prove this conservative is not possible, because if it's a conservative, then you should see two bands, because one would be the N15, nitrogen 15, the other one will be nitrogen 14, all right? For both semi-conservative and the dispersive will show the nitrogen 14, nitrogen 15 hybrid, which is only one band. And so that's why after the collect the DNA, perform the centrifugation, all right, then you see one band, so that proves the replication can be either semi-conservative or dispersive. And then they let the cell continue to grow in the nitrogen 14 media, all right, for 2 generations. And then they collect the DNA, all right, and then they did the centrifugation. So at this time, all right, so for the semi-conservative, at this time, after the 2 generations, then you will see one band for the nitrogen 14, and the other band will be nitrogen 14 and nitrogen 15 hybrid. So you will see 2 bands. And then for dispersive, they will continue to show one band, the nitrogen 14 and 15 hybrid. All right. So after 2 generations, okay, then the scientists see 2 bands here. So that confirms the DNA replication is a semi-conservative replication. Okay, so that's the experiment to prove the semi-conservative replication. And so basically, the replication begin from the original replication, right? And they will open up the double helix, and then they will synthesize in both directions from the original replication. So it's a bidirectional. And at each original replication, there are 2 replication forks, right? So as you can see, this is 2 replication forks, and it's bidirectional. This is an example of the eukaryotic cell, eukaryotic gene, and then there are many original replication. And for bacteria cell, there's only one original replication. This is prokaryotic cell, and then eukaryotic cell. You have several original replication. So the reason they need to have a many original replication is because the chromosome is very long, and then you need to perform the replication, multiple replication at the same time, so that you can reduce the time to replicate the whole genome. And so it's a bidirectional. It doesn't matter it's a prokaryotic or eukaryotic DNA. All right. So they perform the replication bidirectional, and then each. So that's why I say. Each origin replication, 2 replication fork. All right. And so the replication is like a DNA synthesis. And the 3' hydroxy group at the end of growing DNA act as a nucleophile. All right, the nucleophile, this hydroxy group as a nucleophile, because this hydroxy group will attack will attack this alpha phosphate of the incoming nucleotide. And then this after the attack, they will release the pyrophosphate. All right, they release pyrophosphate, and then they will form the phosphodiester bond. Okay, and here just to show you the hydroxy group will attack the alpha phosphate group of the incoming nucleotide, and then release the pyrophosphate. Okay, so that's the DNA synthesis. And then DNA synthesis always a 5' to 3' direction. It's a semi-discontinuous model for the DNA replication, okay? Because when you open up the double helix, one strand is 3' to 5', the other strand is 5' to 3'. For the 3' to 5' parental strand, it'll be simple because you just need to synthesize the DNA 5' to 3' continuously. And we call this strand is a leading strand. And then for the 5' to 3' parental strand, then it's a lagging strand because you have to synthesize the DNA 5' to 3', and then so you cannot perform the 5' to 3' on this strand directly. So you have to do it separately. So you have to about every 1,000 to 2,000 base pair, then they will have the site for the DNA to synthesize 5' to 3'. And so you will have many fragments here. And each of these we call Okazaki fragment. And they synthesize DNA in a 5' to 3' direction. But they are away from the replication fold. For the leading strand, they are going toward to the replication fold. Okay. Yes, Hadel.
Hi, Professor. But they're still getting replicated, though, right?
Yeah.
Okay.
Yes. Both strands are replicating. Yes, okay. Both strands are replicating.
Okay. But over here, why is the replication here is shorter than like the one on top? Like over here, they replicate it right away. Like it's being replicated with non-stop. Over here in the lagging strand.
Yeah. Because the DNA synthesis must go from 5' to 3'. Therefore, the top strand here, there's no problem. They can synthesize the DNA strand continuously without stop, without any pause. But for the lagging strand, because the direction, go this direction, 3' to 5', the DNA cannot synthesize 3' to 5'. Therefore, they have to do it piece by piece, 5 to 3, 5 to 3. They have to about the gap every 1 to 2,000 base pair. They will have a starting point, and then to synthesize this direction, 5 to 3, 5 to 3, 5 to 3. And in the end, then they will use a ligase to repair this gap. Okay, and also this is the simplified model. And then for more details, so you can see this, they have to because this is a replication for it continue to go this direction, and then the DNA double helix DNA continue to open up, so that they have to form this, the clamp that we will discuss in next few slides, the clamp, the replication clamp. They have to make sure they have the same speed as the leading strand, so that they can continue to replicate the DNA simultaneously. Okay, and then of course to synthesize this DNA, all right, you need the DNA polymerase. And then for the bacteria cells, there are three different DNA polymerase. Okay, 1, 2, 3, right? And normally the DNA polymerase 3, right? DNA polymerase 3 will perform the DNA synthesis first, okay? And then later on, the DNA polymerase 1 will come in to repair some missing nucleotide, which we will discuss in detail. That's because the DNA polymerase, all right, DNA polymerase, okay, they cannot initiate the DNA synthesis automatically. They need some RNA primer and then to start the DNA synthesis. And once they finish the DNA synthesis, they have to remove the RNA primer. So after they remove the RNA primer, there are gaps, and gaps need to be filled by the DNA polymerase 1. Okay. So that's the function 1 and 3, and 2 is not studied very well, so it's still a lot of function unknown for 2. Okay. And then these are the protein that required for the DNA replication in E. coli. Okay, so we will go through each of these. Let me see. Yeah, we will go through each of these enzymes. All right, and then in the next few slides. But mainly this is the replication fork and then helicase. The helicase. All right, it's an important one because this one will come in to open up the double helix. And the replication fork is the place where the helicase open up the double helix until the place where you have the synthesized double-stranded DNA. And this region we call replication fork. So helicase will open up double helix. So, and then remember, these are the negative supercoil. And when you open up this, you increase the positive supercoil in front of a helicase. And so that you need a DNA gyrus, which is topoisomerase, to release, to cleave the DNA, to turn it back to the negative supercoil. All right. And then also you need a single strand binding protein. All right. Because this, after you open up double helix, before the DNA synthesized double helix, and you have the single stranded DNA, so you need the SSB to protect the single stranded DNA. And the primase, okay, the primase will produce the RNA primer. So RNA primer, so that the DNA polymerase 3 can initiate the DNA synthesis in the DNA polymerase. And then RNSH, the H represent hybrid. So that means after the replication, the RNSH will come into a degrade RNA primer, okay? And then DNA polymerase 1 will come to fill in these gap, and then ligase will seal the gap. Okay. So this is a DNA gyrus. So as we discussed yesterday, so they will gyrus is class 2 topoisomerase. So they will cleave the DNA on both strands, and then introduce the negative supercoil to the DNA ahead of the replication fork. And then the single-strand binding protein stabilize the single-stranded region, okay? And the primase basically provide this hydroxy group, all right, to initiate the DNA synthesis. And then the DNA polymerase 3, all right, to synthesize the DNA, and then as the replication fork move away, RNA primer is removed by DNA polymerase 1, all right, and in the meantime they replace with the DNA. And the reason DNA polymerase 1 can remove the primer is because they have this exonuclease activity, okay. And then RNSH, so basically they can find these the hybrid and then cleave the RNA primer. Okay, that's the RNSH. And then the replisome here, the replisome, basically the replisome, you have a clamp, you have a sliding clamp, right, to make sure to couple this lagging strand with the leading strands to make sure they move at the same time. So this is the sliding clamp, and we call it the ribosome here, okay, the replication complex. And here's more detail that we have this lagging strand, and then leading strand, and then we have this climb, and so that they can move. This is leading strand here, right, and then lagging strand, so they can move at the same time to synthesize DNA. And the replication pattern, so as you can see here, the original replication, and they've opened up, and you have two replication fork. And the DNA under the control of the original replication, we call it replicant. Okay, and then they open up, we call this a replication bubble. And then they move both direction. And then for the bacteria, okay, for bacteria, so basically you have they move both direction, and then in the midway, then you will see the pattern because the theta. It's a so the yes. And then once they reach the end, there is a telomere signal, and then they will stop the replication, and then they will separate. Okay, so that's for the bacteria. And then this is the example for the phage, bacteria phage. So they have a positive negative strand, so they will nick this positive strand, so 5 prime 3 prime, then they can synthesize from the end of 3 prime, right? So they synthesize at the end of 3 prime. In the meantime, this piece 5 prime end will be peeled off from the negative strand, so that you can see it peel off this and then synthesize the new strand. As this go along, and then finally this positive strand was removed. Okay, the positive strand was removed completely, and then this newly synthesized positive strand complete this replication, and then this original positive blue one, all right, will be separated from this one, and then they will start this replication to complete the negative strand. So this is rolling cycle replication. Okay. So the termination, basically, they have a termination signal here. We call it a tercide, right? So tercide is short 23 nucleotide long, and then they will give the signal, so that they will stop the replication. So the summary of the DNA replication in prokaryotic cell, the DNA synthesis is bidirectional. Okay. The direction of DNA synthesis is only one direction, 5 prime to 3 prime. And then the gyrus basically it's to introduce the negative supercoil for the DNA during the replication, and primase catalyze the synthesis of RNA primer. And the synthesis of new strand is catalyzed by the DNA polymerase 3. Okay, so that's the summary of the DNA replication. Any questions so far? Okay, good. And then this is just the structure of the DNA polymerase. Okay, and so so when you perform the DNA replication, of course, that sometimes they will incorporate the wrong nucleotide. Okay, so therefore the system, the cell system, they have the mechanism to proofread the DNA. And to repair the DNA, okay? Because the DNA replication takes place only once each generation in the cell. And then if there's an error during the replication, that will introduce the mutation, okay? And so that's why the cell develop this proofreading and repair system. And this is just to show you several mutation mechanisms. So they say if it's one nucleotide changed, then you have a point mutation. And then point mutation can have 3 different results. One is a missense mutation. So missense mutation, that means change the one nucleotide from UAU to UCU, then you change the amino acid from tyrosine to serine. Or nonsense mutation. Nonsense mutation, that means you introduce the nucleotide and then that causes the change the amino acid to the stop codon. So you have missense, nonsense, and also you have silent mutation. So that means if you change one nucleotide, then still a code for the same amino acid tyrosine here, then it's called silent mutation. So this is one mechanism that can introduce the DNA mutation, right? It's a point mutation. And then also we have this insertion and deletion. So the insertion or deletion of a nucleotide that can cause the frame shift mutation, because, for example, here is original nucleotide. Then if you add one nucleotide, all right, between this A here, so that you add one nucleotide, then you push this nucleotide to the next open reading frame, all right? So now you have this AGA here. So you have the new amino acid, and then the rest of the sequence change their open reading frame, and that give you a different amino acid. Or the deletion also causes the change of open reading frame. So that's called this kind of mutation is a frame shift mutation. And the tautomer shift. Tautomer shift is just the shift of, like here, all right? Originally this ATGC, TACG. Then during the replication, all right, so you have tautomer shift to the immunoform. So then the A, all right, instead of pairing with T, now pair with the C. And then for another generation, then this C, all right, will bring in this transition mutation, okay? So that's the tautomer shift. So the proofreading basically improve the replication of fidelity. And the proofreading normally for the polymerase 1, okay, catalyzed by the cut and patch, catalyzed by the polymerase 1, and so they can remove the RNA and also the DNA. And mismatch repair, so enzyme recognize 2 bases are incorrectly paired, then the area of mismatch is removed, and then you can replicate that region again. And base excision and repair, so if you have damaged the base, can be released, can be removed by the DNA glycosylate, right, and leave the AP site, and then the sugar phosphate are removed along with the several more bases, and then the pole 1 will fill up the gap. Okay, so this is the polymerase 1 repair. They have a 5' to 3' exonuclease activity, so they can remove up to a 10 base pair, 10 nucleotide in the 5' direction downstream from a 3' hydroxyl single-stranded nick. So that's the repair. And then this is just to show you the proofreading, improve the so this the replication fidelity. So they say you have this the UV light and cause this dimer, right? This dimer, right? And then you have to repair this, okay? So the mismatch repair. So you have the newly synthesized. This is newly synthesized DNA, and then it's a mismatch. All right. So you have mismatch here, G and T, and then you have this mismatch repair. This enzyme, right? It's a mut HSL. They come to recognize this location and then give the methylation of they recognize this methylation strand, the methylation strand, which is the original parental strand, and so they know the other strand. The other strand is the newly synthesized strand. They recognize the parental strand through this methylation site. Okay, so that they know the red one is the newly synthesized, so that they can cleave from the place where they have mismatch all the way to the nearest methylation site, and then they can synthesize the new strain so that they can fix the mismatch. The base excision repair and the nucleotide excision repair. So basically, base excision repair, the damaged base is excised from the sugar phosphate backbone by the DNA glycosylase and creating an AP site. All right, creating an AP site like here. So you have damaged base, and then they remove these. Okay, remove this nitrogenous base, and you keep this sugar phosphate here, and we call this as AP site. Then a purinic or a pyrimidinic endonuclease serve the DNA strand, and then excision nuclease remove this AP. All right. So basically, you remove the nitrogenous nitrogenase base, and then you remove the sugar phosphate. Okay, and then you can have a DNA polymerase, and then to repair this gap, and finally ligase to ligate. Okay, and the nucleotide excision repair. So when a serious lesion, okay, and like a pyrimidine dimer is detected, then the ABC exon nuclease will bind to the region. They bind to the region and cut out a large piece of DNA, including the lesion. Then DNA polymerase I ligase can resynthesize this gap. Okay, so that's a nucleotide excision repair. And then the post-replication repair. So it's like the damage bypass mechanism. All right. So basically, you have pyrimidine dimer that's caused by the UV light. All right. And then you can bypass during the replication. They bypass these dimer. Okay. And then they use the recombination. Okay, recombination to bring to fill the gap. Right. So after recombination, you fill the gap, and then you can have the DNA polymerase I, and then to fill up the gap. And so that's how you perform the post-replication repair. This is a damage bypass mechanism. Then the SOS aeroprone bypass. Basically, in this, if the bacteria subject to the extreme condition, and then a lot of DNA damage occur, then you will have the SOS response. So that we involve the aeroprone repair. Okay. So that's the SOS repair. And that the double strand break repair, that's a DSB. The double strand break repair, basically you have two different types of repair pathway. You have both homologous recombination, HR, and the non-homologous recombination, NHEJ. So basically, these two mechanisms, then they can repair the double-strand break. So here is just to show you the Halliday model, all right, for the homologous recombination repair, all right. So this is if you have nick right here, and then you have a strand invasion. So basically, this pink one will go up there, the blue one connect to the bottom one here, all right. So that's a strand invasion, and then ligation, all right, ligation, and then the branch migration, so that the blue one connect to the pink one, go up, all right, here, and then the pink one, blue one go down, so up here. So that's the homologous recombination. Then the eukaryotic DNA replication. So now it's understood the prokaryotic cell, okay? And we know this cell cycle. We have G1, S, G2, and mitosis. So G1 basically prepare everything for the DNA replication in the S phase. And then after that, after you replicate the DNA, then you can prepare for the cell division. So we can skip this eukaryotic replication. We can skip this. And the telomere, all right. So the replication, it is important for to know that for the eukaryotic cell that when you have a linear chromosome, okay, then the end of chromosome, they have a region called a telomere, because a telomere is a series of repeated DNA sequence that can give the polymerase signal to terminate the replication, okay. So the telomere basically is simple sequence repeat. It can vary from 100 base pair to 20,000 base pair. It's a short tandem repeating sequence like a TTGGG, like a TTGGGG. Okay. And the telomere is important is because and remember for the leading strand there's no problem to continue to synthesize DNA continuously. But if it's a lagging strand, for the lagging strand, then after the replication, okay, then you will produce you will shorten the telomere if you don't have a specialized mechanism to replicate the telomere region, okay. So this is the replication fork for eukaryotic cell, but it's very similar to the bacterial cell, but just a few different things. Okay, it's not similar. So it's like the DNA polymerase, the polymerase here, they can associate with the some protein to form this pCNA, RFC, and to perform the synthesis. So that's the end of the chapter 10. Any question for chapter 10?